Hi there. Welcome to your first day on the job as a plant geneticist. We are in the crossing block of the CLVR soybean breeding program. The supplies you need are in the toolbox beside you. Today, we are crossing different varieties of soybeans. We are attempting to change the composition of the oil in these soybeans to make them healthier for people. We will start with crossing them in the field. Then, we will wait for 10 days before we test the DNA in the lab. So, let's get started. Grab dissecting tweezers out of the toolbox. Our first task is to collect the pollen. Look around this area for the flowers that are most vibrant and purple. They are the flowers that are going to open today. Great, you found one. Use your dissecting tweezers to collect the flower, and then place it into a tube. The tube is closed because it contains a small amount of desiccant, which will help to dry out the pollen. Open the tube carefully so you don't drop it. Place the pollen into the tube and close the lid. Note that the side and the lid are labeled for identification in the genomics lab. Put the tube into the cooler. Start to look for female flowers that have the petals still visible through the sepals. Look for the white coming through. Perfect. That's a good one. Remove the excess flowers from the node now. You need to carefully remove the 5 sepals with your dissecting tweezers. They are small and leaf-like. Use your copper wire tags to label your cross for easy identification later. Use dissecting tweezers to carefully remove the small petals. Anthers will be left. Good. The female part of the flower is the stigma, which is also left. That's where you want to put the pollen onto later. Go back to the pollen that you collected earlier. Use your dissecting tweezers to carefully separate the petals. Then, you have the male part, which is the anthers and filament. It's an oval-shaped structure. Carefully grab it with the tweezers and gently pull it out. Carefully place the anthers and filament on top of the stigma. If successful, this soybean will contain more nutrients and can be grown with fewer pesticides, which makes them healthy and more affordable for consumers. You're all done. We can check it in 14 days to see if we have a successful cross. Use your watch to teleport now. Welcome to the lab. It's been 10 days, so we are going to test our cross. To begin the protocol, we need to put on our latex gloves, and then collect a sample of the soybean leaf that we crossed. Grab a tube for your sample. Notice that this tube is identified and dated as sample 1 on the side and top of the tube for clear identification. Use your sterile transfer pipette with a pipette tip to collect a small sample of the leaf for DNA extraction. Hold the pipette and use your forceps to carefully remove the sample. That's a nice round cutout. Use the forceps to place the DNA sample into the tube. We've just collected enough sample to do the DNA barcoding with. Great work! Always replace the pipette tip before each task to prevent cross-contamination. Place the used pipette tip in the disposal. You're ready to begin with the extraction. Add 100 microliters of the nucleolysis solution by using the blue pipette and a new tip. The lysis solution is on ice and ready to be used. Remember to replace your pipette tip after each task. Add the 100 microliters to your sample, and then place the tip into the disposal. Now you can take the clean pestle and start grinding the sample thoroughly until you see the solution change color. Pestle is Latin for pounder, which can help you remember that the pestle is the pounder. Great work! Use your blue pipette with a fresh tip to add the remaining 500 microliters of the nucleolysis solution to the tube. Add it to the heat block and let your sample incubate for 15 minutes at 65 degrees. Always verify the time and temperature before you start the incubation process. Incubating promotes growth of microbes and is essential in microbiology investigation. Okay, the 15 minutes have passed. It's time to add the RNA solution. It contains enzymes that break down most of the RNA that is present in the solution. Then, the majority of what we get back is DNA. Add the RNA to the tube now. Close the lid and set it in the heat block. RNA is short for ribonucleic acid. It is a carrier of genetic information, a regulator of gene expression, and an enzyme that carries out catalysis necessary for life. Set the heat block to 37 degrees and 15 minutes. Let it incubate for 15 minutes. It's time to add protein precipitation solution. This forces out the proteins that are soluble so we can get rid of them. Put on your PPE goggles because the protein precipitation solution is a bit of an irritant. Use your pipette with a fresh tip to add 200 microliters of the protein precipitation solution to the tube. Notice the milky color change. Put your tube on ice and leave it for 4 minutes. Put your tube into the centrifuge with the hinge of the tube facing outward. This makes it easier to extract the pellet in the next step. Spin it down for 4 minutes. The centrifuge is used to separate particles suspended in liquid according to particle density and size, viscosity of the medium, and the rotor speed. Grab the tube. It's time to remove the excess supernatant. It is the clear liquid that lies above the solid residue after centrifugation. It is also known as supernate. Take your pipette with a fresh tip and suck up as much of the supernatant as possible without sucking up the material at the bottom of the tube. Gently shake the tube by inverting it upside down several times. Take the tube and spin it down the centrifuge at a maximum speed for 1 minute. Remove the tube from the centrifuge and hold it up. You can see the pellet. That's the DNA that has come out of the solution. Open the tube and pour the liquid isopropanol into this liquid waste container. Use your blue pipette with the new tip to add 600 microliters of ethanol to wash away any excess impurities on the DNA pellet. Close the lid and place it into the centrifuge, hinges out, and spin it down for 1 minute. Continue the last process to pour out excess impurities into the waste container. Impurities differ from the chemical composition of the material, so they must be removed. Set the tube down with the lid open and let it air dry for 20 minutes. This is a good time to clean up your workspace and put away your items while you wait. It's smart to have a clean workspace in the lab. That's 20 minutes! You're ready to add the DNA rehydration solution. Add 100 microliters with the blue pipette and a fresh tip. Close the lid and shake several times by inverting it up and down. Then, place it on the heat block at 65 degrees for 45 minutes. Excellent. Remove the tube. Would you look at that? You have DNA that is ready to use. We aren't using it until a later date, so go ahead and place it in the freezer at -20 degrees Celsius. The tube is clearly identified and ready to use later. You successfully crossed a soybean plant and you extracted its DNA for further tests. Plant geneticists will repeat this process and take detailed notes until they achieve the desired traits for farmers to grow a new cross. Great work today!